On behalf of CARTA and my co-chair, Ruslan, I would like to start the Q&A session. The first question is directed to Gunter Wagner. So Gunter, beyond genetics, how can the study of preeclampsia or miscarriage or other pregnancy-related conditions where immune tolerance fails best inform new ways that we can intentionally break immune tolerance in the tumor microenvironment? In other words, besides altering genetic or protein expression, how might this research evolve to explore complex interactions among sugars, lipids, and microbial components that act at these critical interfaces?
That's a big question. It's at least a complex of questions. I appreciate the scope of that question. In particular, the relationship to other diseases that I haven't talked about, but all of them are related to the different ways of how placental tissue and maternal tissue are interacting, and genetic and environmental factors that can influence it. I think at that point there's not very much that can be said particularly specific about these relationships. I think these many of them are not well analyzed at the level of the molecular details, or also epidemiologically, or actually were quite variable results from sort of association studies. So while I appreciate the question, I'm at a loss of saying much very specific about that. Thank you.
Okay, a second question to Gunter. What sort of gene expression alterations to human fibroblasts would buffer them against invasion of tumor cells? And is it likely to include efforts to bovinize human cells, and how might this be achieved?
So experimentally, there is actually a quite large number of gene expression changes that in vitro at least are affecting the ability of human cells to resist invasion. From comparative studies, there's a large number of candidates. In our company, we have actually identified a factor that is very effective, but for intellectual property reasons, I cannot talk about it specifically which one those are. But suffice it to say that there are surprisingly strong effects for specific of those genes that have a large effect on the resistance ability of stromal cells to resist invasion. So there's a big hope that even relatively focused interventions into the gene expression of these cells that can have a major effect on the spread of cancer that can then be clinically used.
Okay, it's back to me, I think, and we have a third question for Gunter. We're trying to concentrate the questions, the early questions, on people who are in a distant time zone, and Gunter is speaking from Vienna, so it's late at night for him, well, at least moderately late at night for him. So Gunter, what does the association between trophoblast-associated expression and vulnerability to cancer mean for susceptibility to cancers between genders in humans? Is there a difference in the expression of genes associated with resistance to invasion between males and females?
This is a very interesting question, and one would expect, and it's quite plausible to expect, that women might be more adapted to resist invasion because they have to have those molecular mechanisms in order to regulate the fetal-maternal interface during pregnancy. Direct evidence that actually males and females differ in their malignancy rates is relatively little, as far as I can tell. I was looking into it a couple of years ago, and there are two difficulties. The one is there's relatively hard to get data that distinguishes between tumorigenesis and malignancy rates, so specifically factoring out malignancy rate from a rate of tumorigenesis, as I have explained, that would be important. And the second is if there are evidence or separation of tumorigenesis and malignancy rates, there are confounding factors. For instance, there is some evidence that women are less vulnerable to skin cancer and die less frequently from skin cancers than men. The problem is that there's a confounding behavioral factor there because females don't expose their torso to the sun less frequently and to a lesser degree than males. And of course, skin cancer close to the vital organs is more deadly than skin cancer, let's say, on the extremities like the legs and the arms. So while this is something one should expect, there is no solid evidence as far as I can tell.
So another question to Gunther. Can you speculate as to what mediates the relationship between skin fibroblasts and endometrial cell types?
Yes, so that's actually an easy answer because the molecular biology gives us a direct avenue to understand this. And the fact is that the cells in our body are to various degrees related to each other and similar to each other. So in particular, let's say fibroblasts from the skin and fibroblasts in the endometrium are both fibroblasts, and they share some of the genetic machinery for regulating genes. And if you have now a mutation that affects, let's say, collagen expression in the skin fibroblasts, the endometrial fibroblasts are also regulating expression of collagen. So a mutation that changes the gene regulatory elements controlling expression of collagen, for instance, has a certain probability to also affect gene expression in other cell types. The same is true for transcription factors and expression that is used in both cell types. So in short, the answer is that this correlation is because cells share gene regulatory machinery, and mutations affecting this machinery will manifest itself in the expression of the genes in both cell types.
Genevieve, this one is for you. Both our knees and our pelvis underwent significant shifts to facilitate bipedalism. So are there any theories as to why knee osteoarthritis is more prevalent than hip osteoarthritis? Is it possible that something in the mechanics of the bipedal pelvis provided a protective measure for the hip that is not extended to the knees?
That's a great question. I mean, I tend to think about things from more of a comparative perspective. And what's interesting with knee and hip OA is that knee OA is more common than hip in both humans and in other non-human primates as well that are not bipedal. So, I mean, there might be some aspects that are protective in humans given our form of locomotion, but I would say that those are perhaps not exclusive protections, since other primates also, when they do develop osteoarthritis, and non-human primates, non-human primates and non-primates as well also get knee osteoarthritis much more frequently than hip. So I think there are perhaps other mechanisms at play than things that are specific to humans. But that's a great question.
Okay, next question is also to Genevieve. Are there intraspecific differences in osteoarthritis rates between captive and wild animals?
Yeah, so in general, what has been observed is that captive animals or not free-ranging animals tend to have higher osteoarthritis occurrence and severity than wild animals. That tends to be true across species where we have data for both. But in general, it's hard. We have limited information across several different species. So yes, it appears that there's more in captivity, but that could be due to many different factors. It could be due to captive animals tend to be older, probably on average. We tend to know their ages better when we can observe osteoarthritis than wild animals, where we tend to classify them as adult, which could be a range of ages. And so there could be some confounding factors on ages being older, and so therefore more OA prevalence and severity in captive individuals. Yeah, but there are also cases of animals that are not wild, but are free-ranging, and specifically macaques have been studied quite a bit at Cayo Santiago, and they also develop osteoarthritis. And so yes, there tends to be a more OA prevalence in captive individuals than in wild free-ranging, but it does vary across species.
Okay, another question for you is, what are some specific candidate environmental perturbations that you might introduce to skeletal cells to study the skeletal matrix degrading phenotype?
Yeah, so what we are doing so far is, I only briefly mentioned this in my talk, but we make little, essentially bone and cartilage cells in petri dishes from different species. And the way that we induce an osteoarthritic-like phenotype is by either exposing them to mechanical stress or to inflammatory cytokines. And those are kind of the two big environmental factors that are thought to induce osteoarthritis in organisms as well. So we, while the actual perturbations are trying to simulate what happens in vivo, they are in vitro perturbations, so they're not quite identical to what's happening in a joint. But those are the primary ways that we induce the cell to start degrading their matrix instead of building their matrix.
Okay, another one for Genevieve. Is there evidence that non-human species with arthritis feel pain from the disease similarly to humans? And if so, is the pain correlated or not with the degree of damage manifest in the bone of the joint?
I mean, there can be hints at pain in non-human animals in kind of observing how they move through the world. If there are changes in how they locomote that suggests that they're having difficulties moving, that can imply that they're feeling pain. But beyond that, it's really hard to gauge in non-human animals because they can't speak to us the way that humans can speak to us. And there's actually a lot of research right now in medical fields trying to understand the links between pain and osteoarthritis, because one thing that I didn't have time to talk about in my talk is that when we think about humans and osteoarthritis diagnosis in a living individual, we tend to only be diagnosed with osteoarthritis if we first feel pain and then go to a doctor, and there's a scan of our joint that suggests that we have degradation. And so if we don't feel pain at that joint, that doesn't mean that we don't have osteoarthritis. We could have osteoarthritis. And when we look at skeletal samples from deceased individuals and from non-human animals as well, we can see a lot more osteoarthritis in those skeletal samples than perhaps what is diagnosed in living individuals. And so there's a lot of trying to understand why and when and how pain develops with osteoarthritis. There's a lot of research thinking about how do neurons grow into the joint region, because they're not necessarily there to begin with. And then as osteoarthritis progresses, sometimes neurons grow into that area so that pain can be sensed, and sometimes they don't. So that's an active area of research.
Okay, our next question is for Barry. Barry Bogan, in your view, what has been the most important evolutionary driver behind humans becoming biocultural reproducers rather than merely cooperative breeders?
I don't have a time machine. I don't know for sure why that's the case. I just don't have a good answer. I'm sure it was competition with for resources with other hominin or hominin-like species, at some time in the past. But the name of the game in evolutionary biology, of course, is have more babies and make sure your babies survive to have babies of their own. And that it was the driver here. Now, why didn't other species of the apes, for instance, or ancestors that became modern apes do the same? Why didn't humans become, bats? I just don't know the answer to those questions.
Next question also to Barry Bogan. What is the role of fathers in biocultural reproduction?
Fathers are very important. I made a joke, I think, during my talk that I said even fathers sometimes take care of babies. But of course, fathers do a lot of important work. Several of my colleagues in CARTA have spoken about fatherhood at other symposia. Fathers and mothers and grandmothers and aunties and everyone else provide important food resources and other material resources, provide social care, contact. The emotional part of biocultural reproduction is just as important as the material parts. So the word love in my more recent writings, and it's two kinds of love. There's the personal love that we have for our children and for others in our lives, but there's also the love that represents the social protection and care that communities give to all the members of that community, of that family, or a community of families. And fathers provide both the material, physical kind of love, but also the emotional kind of love and community love that is essential for good physical growth and mental growth and emotional growth in our human societies.
Okay, our next question is for two of our participants that goes to Barry Bogan and Martin Heusler. In both talks, you emphasized how uniquely difficult childbirth is for humans compared to other primates. A growing trend called free birthing involves giving birth without any assistance based on the belief that it's more natural and the body instinctively knows what to do. Given the biological challenges of human birth, what are your thoughts on this practice?
Martin, are you connected? You go first.
Okay. Well, I think something that is unique to humans is that we all are dependent on some birth assistance. And this is not observed in any of the other primates. There is no assistance at all in any other primates. And this means that we probably also need this assistance. And of course, there's now a trend towards unassisted, so-called natural birth, but I don't know whether this is really natural at all.
I agree with that it's there is no society of which I'm aware today or in the past where women went off and had babies on their own as a typical, normal way of doing things. It's always with assistance. So I don't think it's a good idea.
Unless we accept, of course, that we have a high maternal mortality, and I don't think that is something we can accept today.
Okay, so next question is to Steve Frank. Is there a relationship between degree of invasiveness of the placenta and certain types of cancer risks across species? Are there certain types of cancer that are more associated with this expression of trophoblast-like signatures of certain cancers, more correlated with different species' degree of placental invasion?
Wagner's specialty. I don't know the answer. It's an excellent question. And I think the answer is that there is evidence for that. But Gunter wrote a review article on that. Is Gunter still here?
Yes, I am. Here I am.
Okay, I'm passing that on to you because that's your specialty.
I'm not sure we have the information to actually answer that question, because First of all, the realization that cancer cells and trophoblast cells are so similar is, in my estimation, relatively recent. I'm not sure we know cancer cells from non-humans and non-mice well enough. That is actually a relatively recent development. It actually depends on detailed epigenomic investigations. So a lot of the similarities between trophoblast cells and cancer cells have to do with DNA methylation. And so, you know, so my awareness of this connection between trophoblasts and cancer cells is about 10 years old, but I don't know of any comparative. I mean, that indirect evidence is in the sense that, you know, there are lots of animals that get cancer that don't have placentas and don't have trophoblasts. You know, at that level, there is some evidence that we can use. For instance, it's surprisingly hard to give cancer to a frog. Even with all the most harsh carcinogens that, you know, would be, you know, able to give cancer to a mammal. Frogs don't seem to care for reasons that we don't know. There's also some evidence that there's sort of differences between mammals and reptiles and birds on the other hand. So, but whether that has anything to do with, yeah, I mean, okay. Most of the reptiles and none of the birds have a trophoblast. So this is a very coarse-grained pattern that's related to that. But whether different species of mammals, I'm not aware of that are there's data that would speak to this question.
Let me follow up with a comment on that too, because I think it's interesting. I mentioned trophoblasts, and Gunter, of course, talked about it more extensively, but I think it's important to realize, and this is somewhat speculative, but I believe this is true, that when we talk about trophoblast cellular programs being observed in early stages of lung cancer development, it doesn't mean that those lung cells are acting like trophoblasts and making placentas. What it means is that there are certain gene expression programs which are common in trophoblast tissues, and those subset of gene expression programs can be turned on in a lung cell and transform a lung cell not into a placenta, but into something brand new and weird, which is what cancer is. You don't think of cancer as not a placenta. Cancer is not a fibroblast. It's not any of those tissues. What's interesting about cancer is that it's something brand new that the body is making de novo by co-opting bits and pieces of different developmental programs. And so I think that's one of the really interesting things that's just emerging from the more detailed studies that are being done in single cells.
It's actually historically quite interesting that one of the early, but then you know, not much promoted theories about cancer was that the theory was that it is trophoblast cells that are still, for instance, in women after birth, and that cancers are actually trophoblasts that somehow found their way into the body of an individual. You know, that was a very early theory, but now with the molecular techniques, there's a new connection to trophoblasts emerging that was is totally unexpected.
That one wouldn't have worked too well for men, would it? Okay, our next question is for Andrea Graham. Is there evidence that species with enhanced resistance to septic shock have evolved better regulatory mechanisms for neutrophil activation or for dampening of the cytokine response?
Thank you for that question. It's one that is a burning question that I think about a lot, and I don't think we know enough yet about the distribution of cytokine responsiveness and susceptibility to septic shock across the tree of life, across even just mammalia. And I'm actually on a quest to start sampling that, sampling that variation more extensively. but I agree with the spirit of the question. It's that in principle, there could be a better way to regulate the magnitude of a cytokine response so that it can more precisely match an environmentally determined optimum. In principle, that is possible even given the decentralized nature of the immune system. So I look forward to discovering which species are more clever about it than we might be.
Okay, so next question is to Dina. What factors contributed to the 160 to 40 lifetime cycle difference between hunter-gatherers and Western women from the 1920s? Is there a rebalancing of the same variables responsible for the difference in Western women from a century ago versus today? Or are hunter-gatherers also seeing a marked increase in reproductive cycles over the past 100 years?
Well, I think the main difference in the increase in cycles in Western women is the invention of birth control, right? So we're able to prevent pregnancies, and women in the past were not able to do that. The second question, could you repeat that? A rebalancing?
Yeah, is it just a rebalancing of the same variables responsible for the difference in Western women from a century ago versus today? Or so the end of this question, or are hunter-gatherers also seeing a marked increase in reproductive cycles over the past 100 years?
I don't know that there's any evidence of that. So, I mean, I think the main driver of this is birth control.
Birth control, yeah. So unless they have access to it, they shouldn't be.
Right.
Okay, our next question is for Justin and Erica Sonnenberg. In Erica's part of the talk, there was a graph showing distinct taxa in blue and strains that were present in the Hadza but missing in industrialized populations were highlighted as the main focus. However, there were also strains that were present in industrialized populations but not in the Hadza. Justin talked a bit about how these are generally more oxidative, but is there more to be said or learned about these strains?
Yeah, I mean, that's a great question. And it is interesting that there is this bloom of species that occurs in industrialized Western individuals. Some of them appear to be more or less more oxygen tolerant than what we see in the Hadza, which is an indication that the gut environment in Western individuals has more oxygen than maybe existed in the past. The other difference that we see is some of these species in Western individuals that we don't see in the Hadza are also adapted to host mucus utilization, which we think is connected to the decrease in fiber consumption that Western individuals have in their diet relative to non-industrialized diets. So as humans consume less dietary fiber, the microbiome is forced to rely on other sources of carbohydrates, and host mucus provides a backup source for that. And so some species in the microbiome of these Western individuals are host mucus adapted, which we think is also potentially negative from a health standpoint, because that mucus lining is an important barrier that serves to keep the microbes sort of at a safe distance from the host epithelial tissue.
And just to add to that, I think, you know, in addition to the species differences that the person who asked the question noticed, we also see these functional changes that are independent of species, such as the increase in antibiotic resistance genes present in industrialized populations. So that's another example of a function that's become enriched and reflects this, I think, larger selection that's occurred based on our industrialized environment and the diverse selective forces. We've just ended up with a microbiome that's well calibrated to all the things that we experience in the industrialized world and deviate significantly from an ancestral state.
So has anyone got a startup company that's aimed at producing probiotics specifically using the stuff that the Hadza have that we don't?
Well, you know that yeah, it's an interesting concept, and I think something that a lot of people have thought about. I think that particular instance, you know, there are a lot of ethical issues that come along with that. You know, this kind of idea of bioprospecting, all the samples that we get from the Hadza are strictly for research purposes, and there are groups, and we're working with the Gates Foundation now to write into consent the possibility of using samples going forward for potential health applications, with the idea that there would be stakeholders in those indigenous communities and an ongoing dialogue about how things should be handled. But it is a very sensitive issue. There have been instances of production of either microbes or actually engineering microbes with functions that have been lost as a way of treating diseases. And just a quick example of that, we're one of these what we call vanished species, the species that are going away, is an oxalobacter. So it's really good at degrading dietary oxalates. It's not completely gone. People that maintain it are actually protected from kidney stones because dietary oxalates are a major source of calcium oxalate that form a lot of the kidney stones. And so if you still possess oxalobacter, you can degrade dietary oxalate and you're protected. If you've lost it, it represents a potential probiotic species, as you're saying, that we could bring back and protect ourselves from oxalate precipitating in our you know, kidneys. And so there have been companies that have both tried to make probiotics, other companies that have tried to engineer that function of oxalate degradation into Western microbes. It's an ongoing kind of point of interest. That's one particular function. There are probably hundreds to thousands of candidate functions like this that we need to test and look into.
Okay, so the next question is to Cal Finch. I live about 500 feet from a major freeway, and now I'm concerned. What should I do to protect against dementia?
Well, we all live in the middle of freeway matrix, and at this moment, there is no individual way to protect. We could conceivably wear a mask that would filter out everything greater than 0.1 micron, but that's just simply not practical. So political pressure on the automotive and on the environmental agencies. Thank you for the question, though.
Okay, another one for Caleb. Is there a causal mechanistic description of the effects of gestational air pollution exposure on adult neurogenesis in the mouse hippocampus? Is there any further indication of which receptors or signaling mechanisms are involved?
Grants are pending on that question, and there's a number of points of cell cycle regulation that are potentially targets of existing drugs. So and there may in fact be some overlap with regulation of stem cells in cancer and this compartment of brain stem cells. Open question. Thank you.
Okay, so next question is to Andrea. Phagocytes are considered to be the most evolutionarily ancient blood cell type, and some lower vertebrates have red blood cells with phagocytic potential. Do you think the loss of this potential in higher vertebrates confirms that this behavior has deleterious effects in animals with more complex or tightly regulated immune systems?
I would have to think more and learn more about the distribution of those phagocytic red blood cells to answer the question properly, but I bet there's some really important mysteries to be solved there. So for example, we have more than just neutrophils that are phagocytic cells in our immune system. Quite a number of our different cell types are phagocytic, and potentially as we became more complex and diverse in our cellular types, we could diversify and hone the phagocytic capacities of our arsenal of cells that maybe made us able to give up red blood cells being phagocytic. There's a lot of other differences, though, of course, between red blood cells and white blood cells, including how long they keep their nuclei, right? Because red blood cells evict them. I bet there's a whole lot of interesting comparative cell biology that needs to be done to, and again, across species. I think there's a massive need for comparative immunology to answer the sorts of questions, wonderful questions you all are asking me. Thank you.
Thank you.
Okay, our next question is for Steve Frank. And Steve, thank you for a master class on how to give an engaging talk without using any slides. I wish I'd had that example 30 years ago. Would have changed the way I gave talks.
Thank you.
The question is, even though the placenta is a very proliferative, invasive, and relatively genomically unstable tissue, it is also very well regulated in pregnancy, so its growth is controlled. Which mechanisms could explain how this regulation in normal placentas is lost in different kinds of cancers, besides KRAS and P53, KRAS, P53?
Well, I think that's very similar to the previous question in the sense that it isn't that most tumors are really placental tissues. And the open question, which I don't know the answer to, and again, maybe Gunter knows more about this than I do, is that some of the programs of placentas and trophoblasts can be expressed in what is a lung cell and transform a lung cell into something that's more invasive. And so it isn't, I think that drawing too strong an analogy between the fact that there are evidence that trophoblast-like cellular programs are being expressed in some cells, and then thinking that the tissue is placenta-like is too strong. I think that you have to think of cancer, and this is just a repetition of what I said before, as a pancreatic cell, which doesn't normally invade, doesn't secrete proteases, doesn't do lots of things, doesn't attract blood supply in the way that tumors do. You have to think of a tumor as the origin of a new tissue from scratch. And so my vision, and I think a lot of other people, but what I see is that a cancer progresses by basically taking bits and pieces of different developmental programs and bringing them together to make something new. And so the question is interesting, but I think we need to go in a different direction to really understand how cancers develop. And so what's really exciting to me about modern cancer research is that with new single cell technology, meaning that scientists can now measure the gene regulatory changes that are happening in individual cells, and this has only been available for a few years. And so we can now look at how cells, which are lung cells, pancreatic cells, they're fairly limited in the kinds of gene regulatory programs that they're expressing, and they have the genes to make placentas because the genome of a lung cell, as far as we know, is basically the same as the genome of a placenta. But all the placental genes are mostly turned off on a lung cell. And so what's happening is that lung cell is releasing those brakes and those negative regulators and all of those other cellular programs. So trophoblasts, but chondroblasts, connective tissues. I thought it was interesting that I mentioned that in the lung cells, they noticed that there were expression of tubular kidney epithelial cell programs. So these lung cells are not turning into kidney cells, but tubular kidney epithelial cells are very good at repressing immune responses, and a tumor has to repress an immune response. So I think that the big conceptual leap, if you're trying to think about cancer, is not to think about those tissues, placentas and so on, as appearing in cancer, but thinking about bits and pieces being recombined into something new. This is really the insight that I think we're looking at. Thank you.
Yeah. If I may add a little perspective, is that cell types should be viewed as a mosaic of effector modules, and these modules can be individually redeployed in other contexts. And so the cell type is not a unitary thing. And that is, I think, what you also expressed. So, I think also our view of what cell types are needs to be modified based on the single cell data that is coming out nowadays.
Yeah, so I think it's a very exciting time because I think we're not only are we learning about cancer, but we're really learning about how our bodies develop. And so it's not the same in cancer, but it's the same new insights that are really changing our understanding of both development and of cancer. And that we're really looking at a new era in terms of our understanding because of this new technology. So it's an interesting time.
If I could ask you to add on the epigenomics of cancer tissue formation. It's obvious. I know you've thought about it.
Well, yes, I mean, I think that it's a bit unfortunate that there's such a hard separation between genetic changes, which mean base pair changes in the DNA, and epigenetic changes. What matter in the cells are changes in gene expression, and those can arise both by epigenetic and genetic changes. And so there's a bit of a language conceptual barrier that makes it a bit hard to talk about things, but obviously we're all in agreement. I can tell from your question that I tend to think that there's different timescales over which cellular changes happen, genetic versus epigenetic. Epigenetic tends to be faster. Epigenetic can also be reversed, whereas genetic changes are harder to reverse. So we have basically different timescales of the formation of variation and the transmission of those changes to future time periods. Epigenetics tends to be faster, genetics tends to be slower but more permanent, but they both go on simultaneously. And so a big challenge in any evolutionary study is to understand the simultaneous changes on different time scales, and cancer will be very much like that.
And if I could just add, there's two phases, different epigenetic compartments. One is DNA methylation, and the other is histone acetylation and methylation. And
Yes,
those two compartments have not been fully integrated in people's thinking on epigenomics.
yes. There's, on the one hand, there's a lot known and emerging knowledge of the way different epigenetic changes happen. On the other hand, I think your point is a good one, which is that increasing knowledge hasn't been fully integrated into people's understanding of how things change and evolve literally in cell lineages. But and that's the central challenge, is to integrate that knowledge. There's a lot known, but it isn't well integrated. Again, I think without single cell technology, it wasn't meaningful really. But now that we have that technology, there's a big demand for that integration, as you're pointing out.
Okay, so the next question is to Erica and Justin. How should we conceptualize gut health relative to other domains of health? And how should we interpret the magnitude of its effect, both biological and behavioral, compared to other common health interventions?
Wow, that's great question. Wish we a definitive answer doesn't come to mind for me. I think I think from all the talks we've seen today and all the data we know, there are disease rates that are changing rapidly, and we know that human genetics are not changing rapidly. Epigenetics can change, of course, those may be in play, but I think the gut microbiome and gut health in general, nutrition included, stand as kind of top candidates for driving our modern disease epidemics. And so, I think formal tests of this remain. I think actually changing microbiomes and showing that you can have positive impacts on markers of health for, it's hard to watch chronic diseases develop at different rates in experiments, but I think we can use proxy markers, biomarkers of disease progression as ways to understand how we might causally change gut health and change our likelihood for developing different diseases. So, in my mind, it stands as a top candidate for a lot of diseases. I don't know how you see it.
Yeah, I mean, as a microbiome scientist, I'm probably quite biased, but I would agree that, I think it's playing a major role, especially in the increase in non-communicable chronic diseases that we're seeing in Western populations.
Okay, this one is for Dina. Dina, how are sex steroids increasing over time in women who are not directly taking them? Is this from environmental exposure?
I think some of it probably is environmental exposure, but I think some of it is differences in our diet. So we know that women who have much fattier diets have higher levels of sex steroids. And we know that women who exercise a lot have lower levels of sex steroids. So I think some of it is definitely environmental exposure. But I think much of it is just lifestyle differences.
Are there sex steroids in drinking water?
Ooh. I don't, there was all the concern about, women who take birth control, those steroids getting into the water supply. I don't know the latest research on that, but yeah, I think there's certainly an environmental exposure that we should all be thinking about, but easier to control is what you're putting in your body and how much you're moving it.
Okay, so next question is to Steve Frank. How come evolution has not produced more transmissible cancers that can survive the demise of their host?
Don't know. There's, as far as I know, I'm not an expert on transmissible cancers, but I was talking to somebody who is Murchison in Australia a couple of days ago, and I think there's only 3 well-defined species that have produced transmissible cancers. And I'm not sure if this is true, but I think in most of those cases, the transmissible cancers are relatively old lineages, meaning that there haven't been that many origins, even in those species. Like in the dogs, I know that there was one lineage of transmissible cancer from the 19th century, which I think still circulates and evolves. So these are rare events in evolutionary history, and the hardest thing to talk about in evolution is rare events. There are things that happen in the past, they happen rarely, they have huge consequences sometimes, and trying to say why is pretty hopeless. But our curiosity towards them tends to be great. I mean, to me, the hardest thing about studying humans is that, roughly speaking, there's a sample size of 1. And that's not quite true because there were multiple human lineages at various times in the past, but modern humans, there's really 1 species. And so answering questions about why did humans evolve the way they did is hard. If you have a sample size of 2, you can draw a line. 2 points make a line. You don't even have that. So with the transmissible cancers, I don't know the answer to that, but I would recommend, I forgot her first name, but Murchison in Australia is writing some nice papers on that. So I would point you in that direction. Thank you.
Presumably it's hard to evolve the ability to transmit for cells.
I mean, I was recently pointed to a paper by a student about transmissible cancers in mollusks. So marine mollusks have a surprisingly high number of transmissible cancers compared to our terrestrial mammals. But it's of course much harder to have a somatic cell surviving in transmission.
But it's in a fairly narrow lineage of mollusks. It's not in
well.
mollusks in general, right? So, and I think there are a few origins in that case, but not a huge number.
Not a huge number, but a surprisingly.
Yeah, maybe. So maybe that's, so I don't know, maybe that's an interesting case. I think with dogs, and I don't know, Tasmanian devils, there's only a small number of origins. Maybe the mollusks are interesting, but those are the 3 cases: mollusks, dogs, and Tasmanian devils.
Okay, our next question is for Justin and Erica. Is there any evidence supporting the possibility for moving our industrialized microbiomes back towards those of non-industrialized settings at the individual level, or is that a lost cause? If this is, in effect, a permanent change that cannot be ameliorated, how can we best manage the negative consequences?
I mean, I think there is this issue of whether, even if we were to try to reintroduce these lost species, whether our gut is a hospitable environment for these species to engraft and be maintained. I think there's a lot of evidence that that would probably not be the case. And because the microbiome is so dependent on lifestyle, even if you were able to get some of these species to engraft for some amount of time, chances are, your next course of antibiotics or the next time you're at a McDonald's or what, the different lifestyle choices that we make would make that species not engraftable in a long-term setting. And so the question is, the functionalities that we've lost as a result of industrialization, can we bring some of those back in species or other ways that are compatible with a Western lifestyle? And so that could be species that are able to adapt to our lifestyle that provide some of these functions. And like Justin described earlier, potentially engineering some functionality back into species that we know are capable of inhabiting a Western gut.
And, I think we've over the past 10 years done a lot of dietary interventions here with Christopher Gardner's group, looking at how generally healthy diets and aspects of healthy diets can alter the microbiome and how that can ripple along aspects of host biology, like through different immune markers and things like that. But one thing we monitor in these studies is we do very comprehensive microbiome profiling and never see these lost species come back, even on very high fiber diets, high fermented food diets, things like that. And so it really does indicate that many of these species that we've lost appear to be, at least in the absence of some deliberate reintroduction combined with nutritional support for those microbes, we have irreversibly lost them. And so I think as a first phase, one of the things we're thinking about, we know that there's a distribution of microbiome compositions across people, and there appear to be some people that have healthier microbiomes and some people, there's a distribution, some people that have less healthy microbiomes. Can we take the microbes that are associated with health and create cocktails, administer those to people that have less healthy microbiomes and usually have worse inflammatory markers, and combined with diet, move those people to the healthier side of the scale? So I think phase one is just thinking about how we use the microbes that are at our disposal in the industrialized world to try to get everybody to a healthier spot.
One issue that might come up is you wanted to do something like CRISPR engineering of a bacterium that was common in the industrialized microbiome to introduce the ability to handle oxalate, for example, and try to get general prevention of kidney stones.
Exactly.
The question that comes up is, what is the cost of that? And would it be stable? Could it maintain itself, or would it be enough of a cost to drive it out of the ecosystem? But boy, I'd like to have that.
Completely. And the part of the paper is actually forthcoming. It was just accepted as science, so that'll be coming out soon, that describes that oxalate story. But basically the idea is a lot of these functions that you engineer into bacteria are indeed not adaptive for them, because it's not wired into their metabolism. It's an auxiliary function, but you can engineer in something else, and by providing a nutrient, a privileged niche to that microbe, and providing something in diet that supports them specifically, you can overcome that fitness defect. So the idea would be that you can maintain these microbes that have therapeutic benefit through other means of reinforcing their colonization.
So you need both a prebiotic and a probiotic.
Exactly.
Yeah.
Okay, so next question is to Dina. I'm sorry, it's a long question again, so I'll read it slowly. How might chronic exposures to endocrine disrupting environmental pollutants such as phthalates, PFAS, and heavy metals unintentionally alter the neuroendocrine regulation of menstruation and ovulation? And what do you think might be some of the long-term evolutionary and intergenerational consequences for female reproductive health?
So I think we have a lot of data that suggests that endocrine disrupting chemicals are dangerous for women. So we know that they cause fertility problems. We know that they are causing menstrual cycle issues. So, I mean, I think there's plenty of evidence that suggests that they are bad for women. I think we know the most about how their connection to infertility, less so about just, cycle regularity and other aspects of the menstrual cycle. Long-term effects, I think, just the fertility issues alone are frightening. And, when you let your mind sort of go to the science fiction, you can imagine a time where it becomes, if exposures are very high, you can imagine a time where it becomes very difficult for many women to get pregnant. I don't think we're there yet, so no need to, to freak out. But I do think, fertility, the impact on fertility is well studied and frightening. So.
Okay, our next question is for Andrea. What selective pressures might have driven increased abundance and functional diversity of human neutrophils compared to other species?
There is some comparative work on neutrophil abundances across a bunch of mammalian taxa that suggests that with increasing body size, there's an increased requirement for innate immune cells, such as neutrophils, possibly suggesting that defense of greater surface areas means that cells such as neutrophils are optimal to deploy. So humans do fit into that sort of comparative analysis because we do seem to have higher neutrophil counts than smaller organisms. But again, there's these questions you're asking are right at the edge of the unknown, because I really feel like we need to do a lot more work in these areas. One example of a species about which we don't know enough, remarkably, is Mus musculus, because actually, despite the fact that millions of them are used all the time in biomedical research, an adult Mus musculus, a laboratory mouse, that has only ever lived in the laboratory environment actually is on the verge of neutropenic, not quite neutropenic, but really low neutrophil counts compared to any other adult mammals that have ever been looked at. And that's true even if you account for their body size. And so there's a big movement afoot actually to improve laboratory mouse models by giving them some real world antigenic experience and seeing if they can acquire the neutrophil counts that would place them in line with everybody else. My lab is doing a lot of that sort of work. I didn't have time to fit it into my talk, but one thing we do is we take laboratory mice and put them outside in mesocosms, and we protect them against predation, but we otherwise let them have a lot of real world natural experiences, New Jersey heat and humidity, interacting with soil, all sorts of things. And we see that their neutrophil counts get boosted by that. So I feel like much comparative work to be done, much work to be done to make better use of even our mouse models to answer great questions like you just asked. But my short answer stands that defensive surface area is probably a really important selection pressure in determining what the optimal neutrophil density might be. Caleb, do you have
I could make a brief comment. Our laboratory mice are really different than the native Mus musculus. Steve Ostad and others have compared them, and they're much smaller. They put on weight much less, and their lives aren't increased by caloric restriction like most lab mice are. So it would be worth looking at this neutrophil question in wild-caught mice while they still have the native feral biome, as well as when they're cleaned up from pathogens. And I think that might give quite different answers.
Completely agreed. Yeah. There really are a lot of moves afoot in these directions, I'm happy to report.
Okay, so next question is to Caleb. Would anti-amyloid drugs need to be administered before the onset of diagnosis of dementia?
That was exactly the point of the drug that I showed that was developed by Rudy Tanzi's group to reduce the brain amyloid in people who are at risk for Alzheimer's, for example, ApoE4 carriers in an earlier age group. And there's, my guess is at least 10 such drugs that are in the pipeline. I'm not part of any corporation on that.
Okay, our next question is for Dina. Regarding HYAL2 expression in high-quality embryos, do these enzymes directly digest endometrial polysaccharides?
I think so. So, what HYAL2 does is it basically mediates the breakdown of this high molecular weight hyaluronic acid, I can never say that word, to sort of low molecular weight hyaluronic acid. So hopefully that answers the question.
Okay, so next question is to Andrea. Assuming an abundance of evolutionary trade-offs in the immune system function, like that between pathogen defense and autoimmune risk, how do these trade-offs inform our understanding of modern disease susceptibility, particularly with respect to environmental exposures?
Right. So I think one thing that I was thinking when crafting my talk, but then neglected to say in so many words, is that if the environment of a organism poses different risks of exposure to infection than the ancestral environment, then we end up with the mismatch, right? That this whole symposium is about. I think I failed to say that in my talk. What I think this idea that there's cost-benefit analysis that helps us to understand where that optimum lies, that optimum probably needs, ideally would shift with different environmental conditions, but our evolved propensities to regulate our cytokine responses, of course, cannot keep up. And I think in the Sonnenberg's talk, they alluded to a little bit of this, right? Where microbial dysbioses can place humans with our evolved immune apparatus closer to that catastrophic overshooting zone, where we might be more prone to both acute and chronic inflammatory diseases. Another example that I like to think about is that parasitic helminths are decreasing in prevalence, especially in industrialized nations. And boy, they are really good immunomodulators. They are really good at 24/7 excretion and secretion of a cocktail of molecules that sure reins in a bunch of the danger, reins in the odds of catastrophic overshooting of our immune systems. So yeah, I think this question is absolutely right to suggest that I need to put that trade-off idea in the context of environmental variation, including emerging dysbioses and lack of immunomodulatory symbionts, such as parasitic helminths. So thank you for the question.
I'd like to follow up on that just for a moment. Has anyone calculated the cost and benefit of what the immune system had to do to try to lower infant mortality before there were childhood vaccines, and whether those early life responses of the immune system, which were a great benefit in terms of juvenile survival, were enough to more than counterbalance the late life problems caused by any associated inflammation?
That is yet another burning question. I work with my colleague Jessica Metcalf to try to think about how all the layers of complexity in there, right? Of course, because successful pregnancy, as we've heard over the course of today, does require a bunch of different immunological things to go right. And in the pre-vaccine era, as you've just raised, Steve, maternal transmission of antibodies perhaps even had a even more important effect on early life survival, right? So if you add that in, like how do you immune suppress the mother in just the right ways and at just the right time so that the pregnancy goes to full term, but she can transmit as many of her antibodies as possible to her offspring. That's quite a balancing act there. And then let alone preserve her own life, because a mother who's too immunosuppressed not only makes less antibodies for provision to her offspring, but also she risks her own exposures to infectious disease, right? And then that's bad for both her and her offspring. So all of these complexities make Jess and I want to mathematically model it. So we're trying to approach this from first principles of theory and try to understand where the pinch points really might be for understanding how selection shaped that web of immune steps required for reproduction.
Okay, this is the last question. Steve, how can the conceptualization of cancer as normal development spun out of control benefit the development of cancer treatments? Do you foresee any particular innovations?
Well, I didn't talk, there's a bit older people have been looking at how developmental plasticity is important in the evolution of drug resistance. And so this is a fairly, it's a developing field. 10 years ago, the idea of resistance to chemotherapy was that when you treated with a drug, you had a large population of cancer cells, and because the population was so large, there would be pre-existing genetic mutations that provided resistance. And so basically all you were doing when you treated with a drug was you were picking up those pre-existing cells that had those mutations, and that eventually those cells that carried mutations that made them resistant to chemotherapy would expand and would become the new dominant cellular type in the tumor. But in recent years, people have seen that actually a lot of fluctuation in developmental plasticity is involved in the evolution of drug resistance. I think a simplest example, which maybe I can explain quickly enough, is melanoma. Melanoma develops from neural crest cells. Neural crest cells are a cell that's important in development, which are highly migratory. And this is I think the reason why melanoma is such a dangerous disease, because the melanoma cells have the properties as they differentiate into neural crest-like cells that they are very migratory and therefore they're very metastatic. When people treat melanoma with certain drugs, what happens is that the melanoma cells are fluctuating stochastically between sort of a differentiated melanoma cell and something that's a bit more really like a neural crest. They're not really neural crest cells, but they have more of the neural crest cell program. And what happens with the drug treatment is that this intrinsic sort of stochastic fluctuation, the drug selects for those cells that are in the state that are more neural crest-like, and the more neural crest-like cells are more resistant to the treatment. And so what it means, and then what happens is you get a secondary genetic change which stabilizes the tendency for those melanocytes to be more like neural crest cells and therefore more resistant to treatment. And so instead of there being a pre-existing genetic mutation, what was pre-existing was a exploration of different cellular programs that were natural to a cancer tumor. And then the drug selects for stabilizing certain developmental states which are particularly resistant to the treatment. So I think that this notion of thinking of cellular programs as sort of being the regulation being loosened up, being a more of a developmental creativity, which is normally suppressed, because when you're an adult, you don't want your cells, your lung cells expressing trophoblasts, and you don't want your melanocytes expressing neural crest-like cells. But what cancer does is releases those cellular programs, and so you have this looseness. And then the drug treatments will then often favor some of those subsets of cellular programs. And so this is really helping people to understand why some drug treatments fail or what needs to be done in the cocktail of drugs that are produced and used in treatment. And the vision has changed from we need to deal with the pre-existing mutations to we need to deal with the intrinsic cellular plasticity that a particular tumor has when we figure out how to treat. So that's the simplest answer. I think there are probably other aspects that will come up, but in drug treatment, we see this as is already starting to play a role.
Okay, so I'm just going to say a couple of closing remarks. First, I want to thank all the speakers for a wonderful discussion, wonderful presentations today. We've covered a lot of subjects related to evolutionary mismatch. It's been very stimulating discussion. I want to thank the audience and especially people who provided very interesting questions. And I think as a just a closing remark regarding the topic of today's discussion, I was thinking that we can think of about two types of mismatch. One type that there is nothing we can do about, and that has to do with people in industrialized world with good medical care, just living longer, and because of that, there are increased incidence of aging-associated diseases, cancer, neurodegenerative diseases. These are the things that we just pay for living longer, and there's nothing we can do about that other than just dealing with one disease at a time, maybe. But it's not the battle we can ultimately win. We can just extend it and delay it. But the other type of mismatch is the one that where we could do something about and are doing something about. And these examples of this would be the easiest example would be reduced physical activity due to the fact that we don't have to walk 10 miles to find food or to run away from predators. We don't have that, but we can substitute that with mimics, such as going to the gym. Other examples that we heard about today from Eric and Justin's talk about modifying microbiome, pushing it in a direction that's healthier. And then example that Andrea mentioned, that we meaning people in industrialized world with access to clean water, we don't have parasites, but the rest of the animal world and people who don't have access to clean water all have parasites, and that makes them protected from allergies, for example. We don't want to get parasites, but we potentially could mimic that along the same sort of line of ideas that could be done with microbiome. So there are two types of mismatch, but for most of them, we don't know how to mimic them. We don't know how to mimic parasites to block allergies, for example. So I just want to close with sort of inviting everyone to think about what are other types of mimics of the more natural biological environment that we can think of without sort of paying the price of starvation, infectious diseases, parasitic infections, and all the other things that we got rid of. So, that's all I was going to say. And now I'm going to pass on to Pascal to say the closing remarks.
I want to thank the two co-chairs, Steve Stearns and Ruslan Mejitov, and all the speakers for a really incredible lineup of talks. This question and answer session could go on for hours for me. And I hope you enjoyed this talk. I want to thank the audience. And if you think there is value in this type of exploration, please consider supporting CARTA so we can continue having 3 symposia a year, hopefully with one in person each year. And our next symposium, it will be in person at the Salk Institute here in La Jolla. And the topic of it will be Ancient DNA, New Revelations. And please stay tuned for the lineup for the year 2026. So thank you all very much. That was most interesting, and I really appreciate all your contributions and the good questions from the audience. Bye-bye.